Good morning, everyone. Welcome to Grand Rounds. There's still people jumping on, but I think that we should get started. So I will turn it over to Dr. Jordan to introduce our speaker. Robin, you're up.
Thank you, Dr. Thomas. Good morning, everyone, and welcome to Grand Rounds. My name is Robin Jordan, and I'm one of the internal medicine chief residents this year. It is my pleasure to introduce today's speaker, Dr. Manasi Agrawal. Dr. Agrawal is an assistant professor of medicine at the Icahn School of Medicine at Mount Sinai and a gastroenterologist at the Mount Sinai Hospital with a focus in inflammatory bowel disease. She graduated from the Gandhi Medical College in India with academic distinctions and received the Indian Council of Medical Research Scholarship. She trained in internal medicine at Maimonides Medical Center in Brooklyn and in gastroenterology at Montefiore Medical Center, Albert Einstein College of Medicine in the Bronx. She then completed advanced fellowships in IBD at both Lenox Hill Hospital and the Icahn School of Medicine at Mount Sinai. She also completed a master's in biostatistics at Columbia University Mailman School of Public Health. Dr. Agrawal specializes in the care of patients with IBD, including Crohn's disease and ulcerative colitis. She has publications pertaining to IBD treatment paradigms, outcomes, disparities, and epidemiology. She is interested in IBD prediction and prevention and the links between environmental health and human health. She is also interested in the study of rising IBD in South Asia and other developing countries, and the care of patients from these regions. She's supported by the National Institute of Diabetes and Digestive and Kidney Disease K23 Career Development Award to study early life risk factors for IBD, and by the International Organization for the Study of IBD. Dr. Agrawal is passionate about equity in healthcare and in academia. She has participated in writing guidelines related to IBD care and is involved in committees with the American Gastroenterological Association and the American College of Gastroenterology. She serves as the secretary of the New York Crohn's and Colitis Organization, and she has served on the Crohn's and Colitis Foundation, the Greater Chapter of New York Fellows Committee, and the Chapter Medical Advisory Committee. A very warm welcome to Dr. Manasi Agrawal.
Thank you very much, Dr. Jordan, for this very kind introduction. And thank you to everyone for joining us here today. I hope you can see my slides. So I will get started. So I will be talking about the role of the environment in inflammatory bowel disease, a topic that I'm extremely passionate about. So just in terms of the evolution of inflammatory bowel disease, as we know, inflammatory bowel disease is an immune-mediated disorder. It's associated with dysregulated systemic immune response with predominant inflammation in the gut, leading to bowel damage and tissue remodeling. And this is the part of the picture, this is the part of the spectrum that we see, which happens after IBD diagnosis. And this is when we treat these individuals. We see such patients routinely in the clinic, and in medicine, we see them when they're admitted to our services. However, there is a very long preclinical phase, which we don't see. And this is the phase when in the background of genetic susceptibility, exposure to different factors, which I will talk about, leads to disease initiation, various alterations at the subclinical level, changes in inflammatory cytokines, changes in barrier function, until they eventually lead to IBD diagnosis. However, this part of the preclinical IBD phase is, of course, not overt, and we don't recognize it until we look for it. But this is the part that is key towards understanding IBD pathogenesis, predicting who will get IBD, and towards taking steps for prevention. And this model is applicable not just to IBD, but similarly to other immune-mediated diseases and perhaps many, many other chronic diseases. So what causes IBD? That's the big question. We know that genetics are implicated. NOD2 mutations, IL-23 receptor mutations. Many mutations have been described in very high quality GWAS data. We know that the microbiome is implicated. And again, very high quality studies have been done that have indicated that there is loss of microbiome diversity, as well as loss of altered host microbe interactions, part of which is mediated by genetics. And of course, we know that there are altered immune signatures. There's a great deal of dysregulation in cytokines, various inflammatory pathways, and there's a lot of crosstalk among genes, the microbiome, and the immune function. Talking very briefly about what we know in terms of genetics, this was a very large GWAS analysis, so a genome-wide association study that looked at the genetic markers, the single nucleotide polymorphisms linked with IBD among individuals of European descent and about 1,000 individuals of other descent. So this included Iranian, Indian, and East Asian individuals. And what they found was that across the two diseases, the two subtypes of IBD, which is Crohn's disease and ulcerative colitis, the investigators found that there was a lot of divergence, a lot of heterogeneity between the two populations in terms of the SNPs. So NOD2 mutations and IL-23R mutations were predominant in Europeans, whereas multiple other SNPs were implicated in Asian populations. There was differences in the estimate size, as well as heterogeneity in allele frequency, which tells us that not the same pathways can explain to us what's going on, and not the same pathways lead to risk for IBD in different populations. This was a study of monozygotic twins conducted in Denmark using a population-based cohort study. As we know, monozygotic twins share almost entirely of the germline DNA. And so if the germline DNA is the main factor that predisposes one to get IBD, the overlap between the two monozygotic twins in terms of IBD should be quite significant. However, in this study of up to 13 years of follow-up with analysis of 103 monozygotic twin pairs, Jess et al. Found that the concordance for Crohn's disease was only 63.6%, meaning that about 1/3 of the disease remained unexplained by genetics. When they looked at zygotic twins, it was only 3.6% of the disease that was explained by genetics. So clearly, there is a big piece of the puzzle that remains missing. Genetics explains some, the microbiome and immune systems explain some, but there is a big chunk that we don't know. And we're now recognizing that this big chunk that's not explained by the other factors that we know is our environment, aka the exposome. Now, what can we learn from the history of IBD, which I think is utterly fascinating? IBD was first identified in Western countries, so in Europe and in North America in the late 1800s to early 1900s. The first case of UC was reported in 1859. And since then, IBD incidence has been consistently on the rise in these countries. And when IBD was first identified, this was in parallel with the Industrial Revolution in these countries. And as we know, alongside the Industrial Revolution, there were massive shifts in our environment. There were shifts in the pollution, in access to care, in urbanization, green space, diet, culture, you name it, and there was a shift. Interestingly, at that time, IBD wasn't very much reported in developing countries and recently developed countries of Asia and Africa. But in the mid to late 1900s, when the Industrial Revolution started to shift towards these countries, IBD emerged in the newly industrialized countries, again, alongside shifts in the environment in these countries. And so these shifts, such as pollution, effect of green space, various medications, the loss of hygiene. So the hygiene hypothesis suggests that exposure to infections and parasitic infestations, etc., make us immune tolerant, and too much hygiene, meaning treatment and very hygienic conditions, which is more linked with developed than with developing countries, may be associated with loss of this immune tolerance and make us susceptible to allergens and allergic diseases, as well as immune-mediated diseases. And I'll speak to that some more in a moment, as well as diet. So we know that we've shifted a lot towards processed foods and food additives and so on, which of course is going to affect our gut microbiome with downstream effects. We conducted a population-based cohort study using registry data from Denmark to sort of get a sense of what the incidence and the prevalence of IBD has been over the last 2 decades. So we analyzed these data between 1995 and 2016. And we found that both CD, Crohn's disease, and ulcerative colitis incidences have been rising very consistently. There's a little bit of a greater increase here, but a downtrend, but in general, it has been increasing, and the same goes for CD. When we look at the different age groups and by gender, again, across the board, both CD and UC incidence has been increasing. This here is the adjusted incidence rate ratio, and the number 1 indicates that there is no risk, and anything on the right of 1 indicates that there has been an increasing trend. And you can see here that all of these estimates are quite on the far of 1. And very interestingly, the biggest increase is seen in the pediatric age group in individuals younger than 15 years. And third, we also see increase in IBD in older individuals. So the age distribution of the IBD population is shifting towards an older age. And these data tell us that whatever it is that causes IBD, the genetic factors and the environmental factors are persistent in our environment, and all of these risk factors are ongoing. And so it makes it very urgent to understand what causes IBD and try to predict IBD and, of course, prevent IBD. So moving on to the molecular epidemiology paradigm, the pathway from causes to disease. And when we look at traditional epi or clinical epi, we know that there is exposure, and following the exposure, there is disease. In between the exposure and the disease is a big black box. We don't quite know, for many chronic diseases, we don't quite know what this big black box is. Presumably, it's changes that occur at the molecular level. So the exposure leads to downstream effects, changes at the molecular level, which predisposes one to getting the disease. And now this black box is being looked into some more. And this is the molecular epi paradigm that was proposed by Frederica Pereira at Columbia University, who really is a pioneer in the field. This paradigm is applicable to various chronic diseases, and I propose using this paradigm in the context of inflammatory bowel disease. And I'll walk you through it. So exposures, which are all of our traditional exposures, our environment, our genetic risk factors, name it, these exposures can then lead to the marker being measured in biological samples. So for example, if the exposure is a pesticide, let's say organochlorides, and an individual gets exposed to them, it's likely that these can be measured in biological samples, either in the form of organochloride itself or slightly modified molecules, such as the various adducts that form when they have chemical interactions with different protein molecules. This internal dose can lead to biological effects. And again, I'll talk about that some more. And when they reach a certain dose, which is the biologically effective dose, they mediate downstream biological effects, which can make one susceptible to inflammation, with further preclinical effects, which in the context of IBD can be disrupted intestinal barrier, altered microbe interactions, as well as changes in inflammatory pathways. Until the disease becomes overt, individuals become symptomatic and there is clinically relevant disease, which then the patient presents to the hospital and gets diagnosed with the disease. So between the exposure and the disease, there are multiple steps that take place. And we have to get an understanding of what these multiple steps are so as to characterize this entire causal pathway. And just in terms of summarizing the data that we have on the first and the last events, that is the exposure and the disease. So in IBD, many different environmental exposures have been studied. And here you can see the vast number of exposures that have been implicated in IBD, including many of these from very high quality studies. And these include smoking, exposure to vitamin D, hygiene factors, diet, and so on. But it's also critical to think about the timing of the exposure. And the early life period is very, very important in this regard. The early life period, which is defined from pregnancy, so from in utero to up to early childhood, approximately 5 years of age, is a period of immune maturation, This is a period where organ growth takes place, the microbiome gets established, leading to downstream effects on the immune function, and the immune systems grow. And this is known as the developmental theory of health and disease, developmental origins theory for health and disease. And so early life risk factors are key to establishing health much later in life. And these variables can impact various diseases, chronic diseases, much later in life after a long latent period. And we're now recognizing that this is very relevant in the context of immune-mediated diseases, including IBD. This here is just an example of how early life risk factors mediate downstream effects. So for instance, if we look at suboptimal maternal nutrition, they lead to placental abnormalities, are linked with other adverse maternal risk factors, can affect fetal gene expression adversely, metabolism, and eventually metabolic syndrome, which may not become manifest until many, many years after birth, perhaps as an adolescent or even as an adult. And the impact of environmental factors can take place in many different ways. And these include impact on nerve system function, oxidative stress, epigenetic changes, mitochondrial dysfunction, endocrine disruption, and so on. So the environment really is key in influencing so many organ systems and growth and development at so many levels that it's very important to think about these variables. It is also important to understand and pay attention to the early life period, because intervention during this period is key towards prevention. And so from a public health standpoint, this is really important. When we intervene at the time of the exposure, before the biological effects have taken place, before the preclinical disease has occurred, this can prevent the disease. If we intervene at a later stage, such as during adolescence or adulthood, we may be able to mitigate the disease, but perhaps not necessarily prevent the disease per se. So it wouldn't be primary prevention. And further, if we intervene at the early life period, we can prevent or reduce the risk of disease in future generations, which again makes it very important from a public health standpoint. So to understand better the early life risk factors, we conducted a systematic review to take into account all of the environmental risk factors that have been studied in the context of IBD with exposure in utero and between 5 years of age. And we found over 130 different studies. And I'll walk you through this slide. It's a little bit busy. So across studies, breastfeeding was consistently found to be protective. And this makes sense because breastfeeding is linked with positive effects on the microbiome, a high microbiome diversity, bifidobacteria. It's linked with antibodies or many, many different compounds and substances in breast milk, such as human milk oligosaccharides, which are highly conducive to immune maturation. And so this makes sense from a biological standpoint. There was also a dose-response relationship. So individuals who were breastfed for 6 months or more they were more protected for IBD compared to babies who were breastfed for perhaps shorter durations. And this has also been seen in other immune-mediated diseases. And so this theory holds that the breastfeeding is protective against IBD. We found that exposure to passive smoke, so maternal smoking during pregnancy and during early childhood, as well as various infections, both enteric and non-enteric infections, were consistently harmful, and they were linked with risk of IBD later in life. Whereas immigration and exposure to antibiotics were likely harmful, but these data were not as robust. And then a whole plethora of risk factors, such as birth month, mode of delivery, other perinatal factors, hygiene-related factors. These data were very unclear. And we can understand why this is, because these data are sort of difficult to tease apart using traditional epi studies, and there can be a lot of noise in these studies. And so whether these factors really contribute to IBD risk or not was unclear based on our systematic review. So we decided to delve deeper into it using high quality registered data. Denmark is a small country, but it has very high quality, robust registers that speak to each other using the unique personal identifier. And so we can get data from the birth registry, from the patient registry, from the prescription registry, the causes of death, and so on, and get a lot of different variables over decades, including the entire population. So this really is a very robust design for clinical epi studies. And I have been fortunate to work at a center of excellence in Denmark, which is called PREDICT, which is the Center for Prediction and Prevention of IBD, along with Tina Jess, who is the director of this center. And here is a picture of the center at Aalborg University. So it's really a very beautiful place also to work at. And using these data, we looked at the impact of antibiotics because the data around antibiotics were not as clear. So we delved deeper into this and we looked at what happens when the mother is exposed to antibiotics during pregnancy. What is the risk of IBD in the offspring? And we quantified the dose of antibiotics based on the number of courses. This again is a little bit of a busy slide, but I just want you to see that this vertical line here represents 1, meaning no risk, and any bar that is on the right of 1 is associated with a higher risk. And we found that in the context of ulcerative colitis, 1 or 2 courses of antibiotics compared to none were not associated with a higher risk of IBD. And this makes sense because we know that shorter courses of antibiotics and fewer contact courses of antibiotics disrupt the microbiome, but transiently and to a lesser degree. However, more courses of antibiotics, so 3 or higher, were associated with a 45% increased risk of ulcerative colitis, which again makes sense from a microbiome standpoint, because the more the antibiotics you give an individual, the larger is the depletion of the microbiome and the more sustained this depletion is. And so this holds in terms of the biological plausibility. Very interestingly, we found that this was true in the case of UC, but there wasn't an increased risk with Crohn's disease. And so this helped us tease out the granularity of the data a little bit. We then we conducted a similar analysis addressing the hygiene hypothesis, and we looked at the impact of mebendazole. So mebendazole, as we all know, is a broad spectrum anti-helminthic drug. So it gets rid of all of the helminths, and it's the only anti-helminth or anti-parasitic that's used in Denmark. So we studied what happens when mebendazole exposure happens between the first 5 years of life and what happens to the risk of IBD. Again, we found that mebendazole during the early life period is associated with an increased risk of adult-onset UC, and it increases the risk by 32%. Again, very interestingly, true for UC, but not for Crohn's disease. Why this is the case, we will have to find out, but this fits with the hygiene hypothesis that eradication of parasites is associated with a higher risk of UC. Now, based on this study, whether it's eradication of parasites or mebendazole toxicity itself that leads to IBD risk, we can't separate out the two, but certainly this is consistent with our knowledge of how parasites work and how they lead to immune tolerance, they promote gut microbiome diversity, as well as stimulate type 2 cell-mediated immunity, all of which are conducive towards a robust immune response. We then looked at the impact of immigration. And we first did a systematic review of all population-based studies. And in this, we looked not only at IBD, but other immune-mediated diseases also, such as lupus, multiple sclerosis, type 1 diabetes, and rheumatoid arthritis. And across the board, we found that immigration from developing countries such as India and Iran and Africa to developed countries of North America and Europe was associated with a higher risk of each of these diseases. There were some clusters based on genetics, but overall, this idea that when you shift your environment entirely from a developing country to that of a developed country, it's associated with a higher risk of an immune-mediated disease, that held. We then looked at it more granularly within IBD, again, using the Danish registers, and I'll walk you through this. And we looked at first the first-generation immigrants, so individuals who were born in a different country, and then they moved to Denmark, and we categorized them based on IBD risk in the country that they were born in as low, intermediate, or high. And we found that among first-generation immigrants, the risk of IBD reflected the risk in the country that they were born in. So if the country that they were born in, the risk was low, their risk remained low even after they moved to Denmark. We then looked at the risk in second-generation immigrants, meaning their children. So the parent was an immigrant, but the child was born in Denmark. And compared with native Danes, second-generation immigrants had the same risk. So their risk was as high as that among the native Danes, despite being the offspring of an individual from a different country. This speaks to early life risk factors. So the country that you're born in, the country or the environment that's around you during the early life period shapes your risk of disease later in life. And it fits with the idea that the early life period is really, really important towards immune maturation and the risk of immune-mediated diseases later in life. Moving on to H. pylori eradication, we know again that H. pylori is linked with the hygiene hypothesis, and eradication of H. pylori has been linked with various immune-mediated diseases. And using the Taiwan insurance data, Lin et al. Reported on this, and they found that eradication of H. pylori compared with antibiotic administration for a different reason or H. pylori without eradication was associated with a higher risk of both IBD as well as immune-mediated diseases in general. I will shift now a little bit to a different mechanism or a different methodology for analyzing these data, and this includes the Geographic Information System, aka GIS. And this is a really cool and rapidly evolving technique to understand many different environmental exposures, which cannot be measured using traditional epi techniques. So we can't really quantify air pollution and the green space and biodiversity that individuals grow around based on questionnaire data or register data. But using GIS data, which is satellite data, we can measure street data, the buildings, the air pollution, the green space, and using computational technologies have different metrics, different indices to measure the amount of green space, to quantify green space or biodiversity or pollution, and then integrate that and model that with the risk of disease later in life. And using this technique, using GIS data, Elton et al. Have reported on the risk of IBD based on early life risk factors using the administrative population-based data from Ontario, Canada. So with respect to air pollutants, they found that exposure to different air pollutants during pregnancy was linked with IBD in the babies later in life. And this was especially true for reactive oxygen species. They similarly looked at green space, and they found that, and for that they used the vegetation index, the normalized difference vegetation index, and they found that childhood green space exposure was protective against pediatric onset IBD. This was a dose-dependent effect. So the more the green space the kids grew around, the more they were protected from IBD. And there was a stronger association when you considered kids younger than 10 years of age in the context of disease onset. This holds with the idea of hygiene. This holds with the idea of the importance of biodiversity. But what exactly this mechanism is, that's TBD. So here I put together a directed acyclic graph to depict the hypothesized relationship between green space and inflammatory bowel disease. And what is the mechanism through which this risk is mediated? So it's possible that green space directly leads to downstream effects and affects IBD risk. It's also possible that green space may just be a surrogate for healthy living. So people who live in more green areas, who live more around trees, are more conscious of their health. Their diets may be different, their physical activity, their stress may be different, and that may be the confounder. Green space may mediate through biodiversity. It may mediate its effect through air pollutants. That needs to be delved into deeper moving forward. But these are all the different theories that may be applicable in this association. However, we don't know what we don't know. So if we don't know something, we can't really understand it, or we can't really measure it using traditional epi studies. And this, again, is another directed acyclic graph that I put together. And as you can see here, all of these different risk factors that are implicated in IBD crosstalk with each other. So either they're confounders in pink, or they're mediators in blue, or they may be interaction or other mechanisms of mediation. And so it's very difficult to separate out all of these risk factors and study each one in isolation. Also, no single factor operates in isolation. So for example, smoking during pregnancy is not going to operate in isolation from premature birth or from low birth weight, these factors are all linked with each other, and to disentangle them is very difficult using traditional epi studies, especially when these factors remain unmeasured. There are further limitations of traditional epi studies. As much information and data that they give us, there are limitations which we should be cognizant of. More often than not, these are registered data, so they're retrospective. There's geographic heterogeneity. They demonstrate association, not causation. There's lack of mechanistic data. There are many different kinds of biases, including measurement, reporter bias, etc. And therefore, it's important to shift a little bit towards more objective and more quantitative techniques. And that brings me to the internal dose, which, as I mentioned before, is a measurement of different exposures in biological samples. So this is more objective, and this can be quantified and linked with disease later in life. And now I will talk about some of the works that have been done linking objective data, so internal dose in biological samples, with IBD risk. This brings the concept of omic studies. Now, an omic study, which I'm sure you have heard of, includes measurement of different types of molecules, and these can be transcriptomics, these can be proteins, so proteomics, can be metabolites, so metabolomics, and using very sophisticated high throughput platforms, thousands and thousands of molecules can be measured in a small amount of blood sample. Typically, it uses mass spectrometry techniques, but there are many other sophisticated techniques that can be used. And different kinds of molecules can be measured. This can be done in an untargeted or an unsupervised way, which means that all the molecules that can be measured are measured in the sample. And so this makes it very unbiased and very objective, but also carries limitations. Or it can be supervised or targeted, where we measure a group of molecules of interest. This can give us information about different lifetime exposures. So all of the exposures that I mentioned earlier can be studied objectively and quantitatively using omic techniques. And this can also give us information about different kinds of health phenotypes, so disease types, as well as disease outcomes and phenotypes. And this emerging field is growing so fast, and there's so much work to be done in this space. So in the context of the early life period, we would look at early life cohorts to get biological samples from the prenatal period, from the perinatal period, and early childhood, and study them, conduct omic analyses in these samples to understand risk of disease later. And for that, but many different cohorts have been established, and I will speak about each one of these a little bit. Starting with the MECONIUM cohort, which is measurement, which is understanding the microbiome through a cohort of pregnant women and their offspring. This was established at Mount Sinai in 2014 by my mentors, Inga Peter and Jean-Fred Colombel. This is a cohort of pregnant women with and without IBD. They're recruited during pregnancy, and clinical data and samples are collected during each trimester of pregnancy. Then at the time of birth, more samples, including cord blood and placenta, are collected. And post-birth, we get breast milk samples, and from the baby, we get stool samples periodically alongside clinical data. Many of our babies are now 7 years old, and now they're going to be followed for additional years. The study is primarily run at Sinai. By using the same protocol, we're also recruiting patients in Hong Kong. So this was a really important study that came out of the MECONIUM cohort, and this was led by my colleague, Joanna Torres, who really is a pioneer in the field. And in this study, Inga and Joanna et al. Identified or studied the gut microbiome signatures in babies of IBD moms and compared those with babies of healthy control moms. And this was done from day 7 to day 90 of birth. And they found that as early as that, from day 7 to day 90, there were distinct microbiome differences. Babies of IBD moms had lower microbiome diversity and very specific differences in microbiome signatures. And when these stools were inoculated in germ-free mice, there were impacts on B cell function, as well as on memory B cell function and downstream effects, indicating that these microbiome signatures may be pro-inflammatory, but certainly there are early life differences in the microbiome. This was then followed by a next study looking at fecal calprotectin, which is a marker of intestinal inflammation. And in this study, as you can see here, the red line depicts the fetal calpro in babies of IBD moms, and the green slope depicts that in babies of healthy controls. And there is clear differences. There was a statistically significant difference in the calpro levels between the two groups, which was maintained for up to 3 years of age. That was the duration of the follow-up of this study. Again, indicating that there is evidence of subclinical inflammation in babies of IBD moms. We then looked at the country that the moms and the babies lived in. Did that have an impact on the microbiome? And yes, unsurprisingly, it did. So among babies, both in Hong Kong and in the US, among the mothers, both in Hong Kong and in the US, IBD mothers had a lower microbiome diversity compared with healthy controls. Whereas in babies, this was true in the US. So in the US, babies of IBD mothers had lower microbiome diversity compared to those in the US, compared to those of healthy controls. But this was not true in Hong Kong. In Hong Kong, the microbiome is comparable. This points towards early life differences based on the country that you live in. Again, the early life environment is different, and this demonstrates that the early life microbiome is also different. And this gives us clues and can be explored further to understand the pathway and the differences in different countries, again, emphasizing the impact of the environment. Moving on to another set of samples or biological specimens that we can leverage to understand early life risk factors, and this includes deciduous teeth. So the development of teeth starts taking place during the second trimester of pregnancy, and the deposition of dead teeth and enamel occurs in concentric circles with a very clear demarcation at the time of birth. And so exposures that can be measured in deciduous teeth using mass spec techniques can be separated into prenatal and postnatal exposures. And this is sort of analogous to rings of a tree. This technique has been pioneered and developed by Manish Arora at Sinai in the environmental health group. And using this technique, a pilot study was conducted by Joanna Torres and group to look at the impact of heavy metals on the risk of IBD later in life. So there were 12 children with IBD and there were 16 children without IBD. And then on analyzing the deciduous teeth, it turned out that lead exposure was significantly higher during the prenatal period among babies who went on to develop IBD. This is a pilot study. A much more longitudinal and larger study needs to be done to follow this up, which we are working on. But this, again, emphasizes the impact of early life risk factors, and it gives us these very interesting biological specimens to work with to get objective and quantifiable data on these factors, which otherwise could not be measured. And this is also thanks to this very interesting history in Portugal, or that baby teeth are saved by their mothers. And Joanna, who is an IBD physician in Portugal, was able to leverage this custom to be able to get teeth from babies. So more on that in the coming years. Moving on to another really important source of information, that is dried blood spots. So at the time of birth, in many countries in the US and in North America and in Europe, and in many states of the US, including New York, at the time of birth, within 48 to 72 hours, a small sample of blood via heel prick is collected, and it is saved on a filter paper card known as the Guthrie card. This can be stored for years. And the purpose of this, the primary purpose of storing this blood sample, is in the event of inborn errors of metabolism, or if it is needed to be tested for the baby or for the siblings later in life. But as a secondary purpose, these samples can be leveraged to study these blood samples in terms of omic analyses, and genomics, metabolomics, transcriptomics, epigenomics, and proteomics can all be conducted using these dried blood spots analyses. Which is a really, really important resource, especially since developing birth cohorts, obtaining samples from babies in the perinatal period is extremely difficult. This technique to do an untargeted metabolomics analysis has been developed by Lauren Petrick, who is fortunately also at Sinai. And this is a demonstration of an untargeted metabolomic analysis using which she was able to demonstrate specific signatures that have been linked with acute lymphoblastic leukemia. And we are working on related studies in the context of IBD using the MECONIUM cohort. Now I will shift a little bit to the effects, the biological effects that we can measure, again, in biological samples and study their impact on disease. And as I mentioned before, there can be many diverse biological effects that can take place in response to the exposure and the internal dose. So as an example, I'll talk very quickly about epigenetic changes. An epigenetic change is an alteration in a gene expression without a change in DNA sequence per se. So it can be of many different kinds, the most common one being DNA methylation, in which a methyl group attaches to a CpG site in the DNA. A CpG site is a site where a cytosine nucleotide is followed by a guanine nucleotide in the 5' to 3' direction. And this site is especially susceptible to methyltransferase activity and thereby DNA methylation. And you may have heard of CpG islands, which are very susceptible to these epigenetic changes because these islands have a lot of CpG sites. Various insults such as smoking, exposure to pollution, inflammation, and so on can lead to DNA methylation. And these can be measured using EWAS or epigenome-wide association studies. Other epigenetic changes include changes in histone code, which affect the DNA folding, and in microRNAs with downstream effects based on whether there is up or down regulation of the implicated gene. This is a very busy slide, but the only purpose of demonstrating this is that this slide represents the impact of maternal smoking during pregnancy and the risk of IBD and other diseases later. So using EWAS analysis, investigators in this study identified 69 DNA hypermethylation signatures, all of which were linked to exposure to prenatal smoke. And they linked that with risk of various diseases later in life. Specifically in the context of IBD, this has been studied, but after IBD onset. So in this study, the investigators looked at 100 children with IBD, obtained a peripheral blood sample, separated out CD8 plus T cells, and studied DNA methylation signatures, and found, as expected, that these methylation signatures were quite distinct in individuals with IBD compared to individuals without IBD. However, the limitation of this study is that the epigenetic changes and the disease were measured at the same time point. And so did the epigenetic change lead to IBD, or did IBD lead to the epigenetic change, or perhaps the treatment for IBD? This remains to be determined, and we cannot separate reverse causation based on an analysis such as this. And therefore, it becomes really important to study the preclinical phase of IBD, or for that matter, any chronic disease, so that before the disease actually becomes manifest, it becomes overt, we want to see what signatures are present in biological samples, and this temporality will give us an idea of the causal pathway and the pathogenesis. So it is this period before the IBD, before the disease is diagnosed, that is important in this context. And pre-diagnostic studies before IBD onset are now taking shape, and many different cohorts have been established for this purpose. These include the PREDICT cohort, the GEM project, the Road to Prevention, and so on. I was just looking at the time. So the PREDICT study, this is the Department of Defense cohort, and this was established at Mount Sinai by Jean-Fred Colombel. And in this, what Jean-Fred and colleagues did was, so in the military, all military recruits undergo periodic health assessment, and they undergo periodic biological sample collection, and all of these serum samples are stored. So there's a vast repository of these biological samples. So Jean-Fred and colleagues identified individuals who went on to develop Crohn's disease, ulcerative colitis, and matched them with healthy controls, and then went back in time and did omic analyses on serum samples from these individuals. And the first study that was done was looking at serum samples 1 year, 2 year, 4 years and longer before diagnosis, and various antimicrobial antigens such as ASCA, CBER, OMPC, and so on were measured, alongside a proteomic analysis looking at different inflammatory proteins in these serum samples. And in this study, they found that there were very clear signatures. So these antibodies were detectable before IBD diagnosis, and these proteomic markers, many of which are linked with various inflammatory pathways, were quite altered prior to disease diagnosis. And this graph here demonstrates the area under the curve, which essentially means that it denotes the predictive value of these markers together to predict the risk of disease. So 5 years prior to diagnosis, these markers predicted 76% of individuals who would go on to develop disease, which increased to up to 85% up to 1 year before diagnosis. So these proteomic signatures and antibodies were altered up to 5 years before diagnosis, before the onset of disease, indicating this preclinical period and indicating that these antibodies to microbial antigens and these proteins and different inflammatory pathways were completely dysregulated, and they likely therefore had to do with the pathway from exposure to disease onset. A related metabolomic analysis was led by Ryan Ungaro, who conducted a pilot study looking at the different metabolomic profile, again, among serum samples of individuals who went on to develop CD/UC versus not. And this was pooled with data from the Nurses' Health Study, and they found that bile acid and steroid hormone biosynthesis metabolites were altered. Metabolites relating to various amino acids were altered in Crohn's disease, and metabolites pertaining to fatty acid pathways were altered in ulcerative colitis, again, up to many years prior to disease onset. So this tells us the different pathways that are perturbed, and this gives us a quantification and further insights into the different disease pathways. And also it helps us predict. It can help us develop prediction models to identify high-risk individuals, to identify people who can go on to develop IBD, and thereby the subset of individuals whom we can potentially intervene on to prevent IBD. This is the GEM project, which is led by Ken Krotoru in Canada. And in this study, Ken et al. Recruited 5,000 first-degree relatives of individuals with Crohn's disease. So this is a high-risk cohort in the sense that because they're first-degree relatives, they do have a genetic risk of getting IBD, but at the time of recruitment, they were completely healthy and they did not have IBD. And they conducted analyses to get a sense of intestinal permeability, and they did that using urinary fractional excretion of lactulose to mannitol ratio. And they divided these individuals into two categories. And this is at the time of recruitment, so individuals are completely healthy. They categorize them into persons with normal barrier function and those with impaired barrier function. And they found that when these persons were followed over time, so up to a median of almost 8 years, there was a clear separation between the two. So people who had a highly impaired barrier function were much more likely to go on to develop Crohn's disease, whereas those with normal barrier function remained healthy for the most part. Again, emphasizing that altered barrier function precedes IBD onset for perhaps as long as 8 years, and all of these preclinical changes are many, many years in the making. The last point that I want to emphasize here is that all of these omics, similar to the exposures that I mentioned, all of these omics crosstalk with each other and they have a relationship with each other. And it's important to try to understand all of these, and none of these exposures or none of these omics can be studied in isolation per se. And so for that, we're developing very sophisticated computational techniques, which includes integration of omic data, which can be early, it can be late, integration of different omics data in the same cohort, integration of data in diverse cohort, and considering how very, very large these data sets are. This is computationally extremely challenging. Having said that, a lot of work is being done at a very rapid pace in this space. And so this is a very exciting time to be doing omics analysis so as to separate out in granular detail the different risk factors and the different pathways that go from exposure to IBD. And again, this is applicable to different immune-mediated diseases as well as other diseases. So in summary, to put this all together, various exposures lead to alterations in metabolites or alterations in proteins or metals, and they can be measured in biological samples. They lead to downstream effects such as DNA hypermethylation, endocrine disruption, changes in oxidative pathways, followed by preclinical effects such as intestinal inflammation, dysbiosis, changes in permeability, until they become manifest as overt disease. And understanding these pathways help us understand IBD pathogenesis, develop therapeutic strategies, predict who can get IBD, but most importantly, from a public health standpoint, can help us develop strategies towards preventing disease. And as the very last point that I would like to make is that I hope I have convinced you that how important our environment is, and we're increasingly recognizing the footprints, our footprints, how they affect the environment and how the environment affects human health. Human health is very, very closely linked with environmental health. And so more research in this space, more awareness in this space is key towards improving both human and environmental health. And with that, I'd like to thank all of my colleagues, friends, mentors, collaborators, especially Jean-Fred, Inga, and Tina Jess, all of the amazing cohorts I get to work with and the funding agencies. And I thank you for your attention, and I'm happy to take any questions. So thank you.
Thank you, Dr. Agrawal. That was really terrific. I think we do have some questions in the chat.
Okay.
So let's see. So Dr. Sacker put some in there. He's 3 questions. So what have we learned from investigation of disease clusters among unrelated individuals?
So diseases, so Dr. Sacker, do you mean diseases like different immune-mediated diseases clustered within the same groups of individuals, or separating out the genetics?
I'm referring to those reports of neighborhoods or farms or things like that where non-consanguineous neighbors and relatives have come down with a very highly unexpected cluster of IBD.
Yes, that's a great point. And thank you for bringing that up, because this helps us. So because they're not related, they don't share genetic material. But because they live in the same environment, they do get exposed to similar amounts of green space and pollutants and biodiversity, perhaps may have similar patterns in terms of culture, social life. And that emphasizes the impact of the environment on the risk of disease later.
Exactly.
Yeah.
And another question from Dr. Sacker is, if maternal transmission is important, we would expect greater risk among offspring of IBD mothers than among offspring of IBD fathers. Have studies of this question provided any definitive answers?
That's also a great question. And I think there are studies that have indicated that transmission from IBD mothers is a little bit higher than that in IBD fathers. But based purely on genetics, you would think that this is not the case. But it's possible that IBD mothers, in terms of the mitochondrial DNA, there are differences. And then in terms of transmission of the microbiome, there are differences. So an IBD mother is more likely to transmit the dysbiotic microbiome than an IBD father is. And so all of these variables need to be taken into account. And I think your third question is, any differences in smoking risk between CD of the colon versus small bowel? I'm not entirely sure. I think there is. I think Crohn's colitis is linked with smoking more so than small bowel Crohn's disease. Or it's the flip. It's the other way around.
Right. It's the risk profile of Crohn's disease of the colon more closely resembles that for ulcerative colitis than for Crohn's disease of the small bowel, emphasizing the really important difference between the two forms.
Right. Yeah.
Nice. Thanks, Dr. Sacker. There's a question here. What is your opinion on the use of vagal nerve stimulation that's shown to reduce level of inflammatory markers like TNF alpha in the management of IBD?
That is a great question. This is also an evolving space, and a lot of research is being done on this. And I think it is being led by a researcher at North Shore in terms of vagal nerve stimulation, and also in Europe. And the more we learn about it, the more we recognize that this may be an important factor. And we're now increasingly, sort of relatedly, but a little tangential, we're recognizing the overlap between IBD and cognitive disorders. So individuals with IBD are more susceptible to developing Parkinson's later in life. IBD is also being linked to dementia. So certainly the nervous system and the gut are linked. The gut-brain barrier, we know about that. So the short answer is TBD. It's not entirely clear, but I do think that the influence of the nervous system, including the vagus nerve, as it relates to inflammatory pathways, is going to be very important.
Nice. Another question. Has anything distinguished the predict cohort with positive markers who got IBD from those with similar positive markers who did not, parentheses, yet get IBD?
Yes. There are, and I did not present some of these data because they're still being worked on and in the interest of time, but yes, there are data on different glycomic signatures, as well as GM-CSF antibodies and other antibodies, which are more likely to be present among individuals who go on to develop IBD versus not.
Gotcha. And then Dr. Golko has a first to comment, amazing presentation. I agree. You said that 60% of susceptibility seems to be accounted for from genetics. Can you calculate individual genetic risk, and how much more can be predicted by adding early life antibiotic exposure?
Yes, I think that's a great question. And I wouldn't be able to give you an estimate quite yet, but we are working on this. We are working on developing prediction models based on early life risk factors. So we're going to be including the different factors during the early life period, linking that with genetic risk to identify how we can predict risk and how much of this can be attributed to each variable by itself. So I will keep you posted on that. Thank you, Percio.
Nice. That brings us to our last question. The end of the question brings us to the 9:30 hour too. So really, Dr. Agrawal would like to say thank you very much for an excellent presentation. Really great to see all the terrific work you're doing. So thank you for all that. Thanks for being a role model in all this. Have a great day. Yes, absolutely. Have a great day, everyone. Enjoy. Thank you.